myself.
Yeah. Okay.
Okay. I'm going to talk about the role of capillary pericytes in controlling cerebral blood flow in health, ischemia, and Alzheimer's disease. And I'm going to start out by looking at why and where brain energy supply is controlled. I'll show you that pericytes on capillaries can constrict and dilate these vessels and look at the mechanisms which regulate their tone, that's sources of calcium and the synapses controlling that calcium. I'll then bring some glial cells in, as most of you are from this glial cell club, looking at the role of astrocytes. And I'll show you that there's different signaling to capillaries and arterioles. I'll then turn to pathology and look at the role of pericytes in ischemia, in Alzheimer's disease, and briefly consider some therapeutic approaches. So let's start out looking at why brain energy supply is controlled. If you look at the brain of a typical Donald Trump voter, you will see that 2% of it, 2% of his body is the brain, but that uses 20% of his resting energy. And because of this massive energy use, the brain is stuffed full of blood vessels, bringing in energy in the form of oxygen and glucose. Now, those of you who've worked, for example, on brain slices, like this hippocampal slice, where these little bobbly things are the neurons, will be familiar with what the neurons look like, but you may be less familiar with the fact that to power the hippocampus, you have this dense network of blood vessels. But even then, you can't have all of the neurons working all of the time. If some neurons up here want to think about something, maybe where the body is in the world, they need to increase their local energy supply in order to function. And they do this by releasing dilating factors, which make the local blood vessels larger, and that brings in more oxygen and glucose. And that's part of what my talk today is going to be about. So where is brain energy supply controlled? If you think about the cortex, blood comes down from the surface of the cortex through penetrating arterioles and goes into capillaries, which provide nutrients to the neurons. And the dogma used to be that most energy control is done at the level of the arterioles by the smooth muscle adjusting the diameter of the arterioles and hence the blood flow. But it turns out to be more complicated than that, because these guys a very long time ago, one in Germany and one in France, discovered these cells which turn out to be contractile, at least some of them are, on pericytes, and they can also regulate blood flow at the capillary level. In fact, these cells have got 5 different functions at least. They're involved in the formation of blood vessels, they maintain the blood-brain barrier, they help to form the glial scar in spinal cord injury. They regulate immune cell trafficking. And what I'm going to talk about today is that they control capillary diameter. Can you follow what I'm saying, or am I speaking too fast? It's okay? Okay. So I just want to spend a minute on the rise of studies on pericytes. As I said, they were discovered more than 150 years, nearly 150 years ago on the left side of this graph. And they were named 50 years later, but pretty much nothing was published on them for about 100 years. But now there's an exponential increase in the rise of publications on pericytes. In fact, one paper on pericytes is published every day, but they remain seriously understudied. In the brain, they're present at 30 to 50 micron intervals along the capillaries. They're quite a major cell population. But if you go to PubMed, you'll find that only one paper on the brain in 1,000 mentions pericytes. And it's even worse in the heart. In the heart, they're the second most numerous cell type after endothelial cells. The myocytes are an almost irrelevant third, but only one cardiac paper in 5,000 is about pericytes. So clearly we're missing out on some therapeutic opportunities because pericytes contribute to all diseases involving ischemia, such as stroke, as we're here today, heart attack, spinal cord injury, as well as Alzheimer's, which I'll talk about today, and some other diseases. But they're largely ignored by scientists and big pharma. So I'm going to try to persuade you to work on pericytes. This is what they look like. If you look in the cerebellum or the retina, on these green capillaries, which are labeled with fluorescently tagged isolectin B4, you can see these red cells here, which have been labeled with antibody to the proteoglycan NG2. And if you look at them closely, you can see they put out long processes along the vessel and around the vessel if you patch clamp them to put a dye into them. And so they seem designed to squeeze the vessel and adjust its diameter. And it's interesting that Edith Hamel has shown that 2/3 of the noradrenergic innervation of the vasculature in the brain is of capillaries, not of arterioles. And so that maybe that's telling us something about their importance for regulating blood flow. So I want to start out by showing you that pericytes can constrict and dilate capillaries. This is a capillary in the retina, where the retina is good because it lets you see a large flat area with the capillary all in focus. And there's a pericyte here we're going to depolarize with the patch pipette. And then there's one here as well and one here. And you can see that when I depolarize the pericyte, it squeezes the vessel and in fact occludes it completely. I'm now going to play that again, and you'll see that after this one has squeezed, this one down here squeezed as well. And so the pericytes can talk to each other. After this one constricts, this one down here constricts, and that's done by an electrical signal going along the endothelial cells. So if you plot the diameter at this central capillary, this central pericyte here, and these ones on each side, then you see you get a repeated constriction of the diameter. Diameter is plotted here against time. That also happens for the other two pericytes, but there's no constriction in between. And that shows you the only contractile cells on the capillaries are the pericytes. Now, how does this happen? If you repeatedly evoke this calcium away, you can see that you can abolish the constriction just here when there's no calcium outside. So they're just like little muscle cells. The constriction goes down to 0 in 0 calcium. And there's also a poorly maintained dilation when you take the calcium away, as though they're producing some constant tone, which is lost when you take the calcium away. So what's happening here? Let's look at the mechanisms producing this tone. Let's look at calcium sources first of all. If this is a pericyte with its little arm around a capillary, then what I've done in the experiment just now is to put a pipette onto it and depolarize it. That opens voltage-gated calcium channels, which let calcium in, and that activates myosin light chain kinase, puts a phosphate on the myosin, and that squeezes the capillary. So this is just like ordinary smooth muscle. But there are other ways to raise the calcium. If you apply an agonist for contractile G-protein coupled receptors like endothelin, which is probably the most powerful vasoconstrictor in the body, if you measure calcium inside the cells, you can see that when you put endothelin on, it goes up. This is in a cortical vessel. So you see the calcium goes up here, which it doesn't do if you just apply ACSF. And at the same time, if you measure the diameter of the vessel using a calcium-independent co-expressed dye here, you can see that the diameter goes down here compared to here. So a rise of calcium produces a constriction. And so what's happening here is that endothelin activates a receptor which generates IP3, and that dumps calcium from intracellular stores, and that again evokes a constriction. So there are these two ways to raise the calcium inside the pericyte. So where is blood flow control initiated? You might think that it's done by the smooth muscle, but I'm going to show you today it's done by the pericytes. And a good reason for that is that most neurons are closer to pericytes than they are to arterioles. They're about 15 microns away from a capillary, but about 100 microns away from an arteriole. So if you're a neuron and you want to adjust your energy supply, one way to do that is to signal to nearby pericytes. They can then propagate a signal along the endothelium, telling other pericytes to change the diameter, and that may go on to the arteriole as well. Why do we want to know this? Well, we want to know, first of all, whether pericytes can also be constricted or dilated by messengers released from neurons or glia. That's what I'm going to show you in a minute. And this matters because we want to understand the normal control of blood flow, how blood flow changes in pathology, and also how functional imaging signals are generated. You will all have seen these nice pictures of orange blobs superimposed on structural images of the brain. And these are known, this is the amygdala responding to a scary face. these signals reflect an increase of blood flow, which decreases the local concentration of deoxyhemoglobin. And so the question is, what increases the blood flow? And I'm going to show you today that it's done mostly by pericytes. So we want to understand the generation of the bold signal. So let's look at the calcium sources and synapses, which I'm going to turn to synapses now, which regulate this tone. Noradrenaline in the brain is released by the locus coeruleus axons. And if you measure the diameter of one of these capillaries and apply noradrenaline, you get a constriction of the vessel. And if you then superimpose glutamate to mimic neuronal activity, you get a dilation of the vessel. I'll show you a movie of that happening in the cerebellum. This is the cerebellum molecular layer. This is a capillary with red blood cells in, and there's a pericyte just about here. And when we apply noradrenaline, you can see in the graph at the bottom, you'll see the pericyte squeezes the vessel, then superimposing glutamate dilates it again. Oops, how do I get out of this? So what generates this capillary dilation? Well, here's a typical glutamatergic synapse in the brain. 85% of your synapses are glutamatergic like this. And of course, you're all aware of glutamate acting on AMPA and NMDA receptors to do signaling in the brain. But glutamate also can act on astrocytes in young animals through metabotropic glutamate receptors. And in old animals, presynaptic glutamate release evokes a calcium change in another way, which I'll show you in a minute. And these calcium changes generated at synapses are very important for regulating the energy supply. And that's because if you do an energy budget in the brain, like Simon Laughlin and I did about 20 years ago, you find that synapses use more than 50% of the brain's energy. And so it's appropriate that they generate the messengers which regulate the brain's energy supply. So glutamate raises calcium by NMDA receptors in neurons and by G protein coupled receptors and ATP receptors in astrocytes. And these calcium changes activate phospholipases and nitric oxide synthase, which generate messengers like nitric oxide and prostaglandins, which serve to dilate the blood vessels. And they do this in the context of a background constriction produced by noradrenaline release from the locus coeruleus. So how do we know all this? Well, we can patch clamp pericytes. This is a pericyte expressing DS-red in a transgenic mouse. We can then patch it with a lucifer yellow fill pipette to make sure we patch the right cell. And if we then apply glutamate or NMDA, or if we stimulate axons running by it, we get an outward membrane current produced. This is again in the cerebellum molecular layer. This is glutamate release from the parallel fiber axons. You get an outward membrane current produced, which is blocked by TTX. Its action potential evoked in this case. And the size of this outward current will generate a 9 or 10 millivolt hyperpolarization of the pericyte. Now, normally, of course, when you apply glutamate to cells, it generates an inward current, not an outward current, because it's opening cation channels with a reversal potential around 0 millivolts. What's actually happening here is that glutamate is releasing a messenger, it turns out to be prostaglandin E2, as I'll show you in a minute, which opens potassium channels. And these potassium channels generate the outward current and hyperpolarize the cell. Because the resting potential of these cells is at about minus 40 millivolts, they have a tonically activated L-type calcium channel, which is letting calcium into the cell all the time. When you hyperpolarize them, you turn off that calcium channel, and that will, we predict, that should make the cell stop contracting. And some indication that's true is that if you measure the calcium in pericytes in vivo, when you stimulate the whisker pad of a mouse, which has got calcium dye expressed in its pericytes, you find that there's a decrease of the calcium concentration in the pericytes when the neurons are active in that region. Okay, and similarly, if you then take a piece of cortex and stimulate axons running across the cortex and image capillaries here, you find that the diameter of the capillaries gets larger when you stimulate the axons. Now, to do this experiment in a brain slice, you have to first of all pre-constrict the pericytes, because by making a brain slice, you've removed the noradrenergic input from the locus coeruleus, which is normally keeping them nice and constricted. So to replace that, we either apply noradrenaline or a thromboxane analog called U46619, which acts on a receptor on the cells, and that induces a constriction. This is diameter down here at the bottom. So the diameter gets smaller, and when we stimulate the axons and release glutamate, the diameter gets larger again. Okay, so using this approach, you can do some pharmacology to see how this works mechanistically. This is blocked by TTX, as you would hope, because it's generated by action potentials. It's also blocked by an AMPA receptor blocker, showing that postsynaptic activity of neurons is needed. But surprisingly, it's not blocked by an NMDA receptor blocker. And this really surprised us because we thought this might be generated by a rise of calcium in postsynaptic neurons, but it's not, and I'll come back to that later. So we wondered whether astrocytes were involved in this, because there have been suggestions in the literature, notably from Brian Movica's group, for example, that astrocyte calcium rises might regulate blood flow. And so to look at this, we do a simple, in principle, experiment where we buffer the calcium in the astrocyte network. And so we do this by attaching a patch pipette to the cell. In the patch pipette, there's either a high concentration of a fast-acting calcium buffer, like Bacta, or there's a low concentration of a slowly activating slowly acting calcium buffer, like EGTA. And that then goes into the cell, and the idea is that if you've got the Bacta there, it will buffer the calcium and stop the release of this messenger, which is going to dilate the capillaries. So here's the experiment being done. This is a couple of capillaries in a cortical slice. There's an astrocyte under here somewhere, which you can see we filled with a green dye, and it sends an input onto this capillary over here. And we then do the following experiment. We pre-constrict the capillary with this thromboxane analog, and we then patch onto the astrocyte with a low concentration of a slow calcium buffer. And when we stimulate the axons to release glutamate, then you get a large dilation, okay? Then you can do the experiment again, exactly the same, except with a high concentration of a fast calcium buffer in the, going into the astrocyte. And in this very carefully chosen example, when you stimulate the axons, then you get absolutely no dilation produced of the capillary. And if you do this over and over, in lots of vessels, you find that on average, having Bacta inside the astrocyte network reduces the dilation by about 70%, something like that. And you can then do other pharmacology, which I'm not going to bore you with, and you find out that the sequence of events is as is shown here. This is the astrocyte, and this is supposed to be a synapse near the astrocyte. We believe that what happens is that excitation of the postsynaptic cell releases ATP, and that acts on P2X receptors on astrocytes to raise their calcium. We can monitor this with a calcium dye. And that then activates a phospholipase, which eventually generates arachidonic acid, and that then generates prostaglandin E2, which acts on EP4 receptors on the pericytes. And any of the blockers of these different enzymes or of this receptor will prevent this dilation, which is produced by stimulating the release of glutamate. Okay, so we think what happens then is that glutamate is released from neurons. It acts on postsynaptic neurons, which release ATP, and this then raises the calcium in astrocytes, which releases prostaglandin E2, and that dilates the capillary. So we sent this off to a nice journal, and they said, well, that's all very fine, but we'd like you to do it all again on arterioles. Why are you so obsessed with capillaries? So we did it all again on arterioles, and it turns out it's completely different for the arterioles. In the arterioles, what happens is that none of the blockers of this astrocyte pathway have any effect, but instead, blocking NMDA receptors or blocking nitric oxide synthase prevents the dilation of the arterioles. So we think we think there are two separate pathways regulating blood flow in the brain here. One is an astrocyte-mediated pathway, which controls pericyte regulation of capillary diameter. And the other is this NMDA receptor nitric oxide pathway, more familiar to many people, which controls the dilation of arterioles. So we want to know what happens in vivo, because lots of cardiovascular type of people don't like the idea of doing experiments in brain slices because they argue that there's no blood pressure, there's no blood flow, lots of key things are missing. Why would we possibly get the right results looking at it there? And so to get around this kind of objection, we look in vivo. This is a diagram of the plumbing of the cortex in vivo. So peel arteries, arterioles, which are very large actually, go down into penetrating arterioles, which are about 10 microns across with smooth muscle around them. They then branch off into different order capillaries, like first order, second order, and so on. And we use 2-photon imaging to image simultaneously the dilation of arterioles and the dilation of capillaries. And what we wanted to show was that capillaries can dilate independently of arterioles. And we do that by showing that they actually dilate first. And that means that it's an active relaxation of the capillary that's happening and not, they're not just being pushed open because the arterioles dilate. So this is looking down into the head of a mouse where there's a green dye, phytidextran, in the blood, and the pericytes are red because they express Mg2-DS red. This is a promoter which exists in pericytes. And so this just here is the penetrating arteriole. This is the first order capillary, the second order capillary, and so on. And these black lines are made by red blood cells going by. And if we stimulate the whisker pad on these animals, what you find is that all the vessels in this, or about half the vessels in this field of view, will dilate rather quickly, just consistent with the bold response being very quick. So if you look at what we call the north order vessel, which is the penetrating arteriole, about 60% of those dilate. And it's similar for the first order capillary. And then there's a gradual drop off as you go further into the capillary bed, which correlates with the fact that pericytes further out in the capillary bed express less contractile proteins. Now, if we focus in on just an arteriole and a capillary, what we find is, as you can see in this graph at the bottom, which gives away the answer, the capillaries dilate first. Okay, so I'm going to play the movie, and I want you to look just here near the capillary, first of all. When we stimulate, the capillary dilates, and only later the arteriole dilates. In this particular example, the arteriole dilates very slowly compared to the capillary. I'll play it again. So here, the capillary dilates, and only later the arteriole dilates. So this tells us it's an active relaxation of the capillary by the pericytes relaxing. Now, so who cares? Well, it turns out it really matters. If you look at the dilations produced in these different sizes of vessel, the penetrating arteriole, the first, second, third, and so on, order capillaries, it's about 6% in the arteriole, maybe 8% in the first few capillaries, and then it drops off again. But on average, the capillaries and the arterioles dilate by about the same amount. Now, it turns out that David Kleinfeld's group at UCSD have quantified the topology of the cortical vascular network. And they worked out that if you go down a penetrating arteriole, through all the intervening capillaries and back up a venule from layer 4, then the resistances in those three parts of the vascular network are in the ratio 1:4:2. So 4/7 of the total resistance is in the capillaries. And what's more, the venules don't change their diameter. So actually 4/5 of the adjustable resistance is in the capillaries. And plugging in our measured dilations, we estimate that more than 80% of the increase of blood flow is generated by the capillaries. So if you look at this diagram of the plumbing again, active neurons and their associated astrocytes release stuff, prostaglandins, which dilates capillaries. That signal may propagate along the capillary. And the capillary dilation dominates the decrease of resistance to flow, and it is also the first thing that happens. So it's faster and it's bigger in terms of its effect on blood flow. So on the whole, the capillary dilation by pericytes is mostly what is increasing the blood flow. So capillary dilation also dominates the bold response. And what that means is that when you see those orange dots on those pictures those psychologists show you, you should be thinking pericytes. Okay. Right, so what I've shown you so far then is that brain energy supply is important to be controlled because the brain uses lots of energy. It's controlled at the capillary level by pericytes. Pericyte calcium is a key mediator of constriction. This is regulated by astrocytes, and there's different signaling to capillaries and arterioles. I now want to turn to the dark side of it and talk about the role of pericytes in various diseases. But if people have any questions on the first part, I don't know if you want. No, I'm not allowed to take questions now. Okay, I'll just go straight on. You're muted, Jorge, so we can't hear you. Okay, I'll just go straight on.
There is one question.
There is?
Yes. What is the percentual contribution of capillaries and arterioles, respectively, in the vasodilatory responses? So the contribution
to the vasodilation, or did you say what is the contribution
to vasodilatation responses?
I think the capillaries produce about 80% of the increase of blood flow.
Okay.
Does that answer the question?
Yeah, I guess. Yes.
You can ask it again at the end if it didn't.
Okay, perfect. Okay.
So let's go on to pathology. Some of you may live in big cities. This is the diagram, a diagram of the incidence of stroke as a function of age in a big city. It happens to be Melbourne, but I'm sure it's the same in your own big city. And you can see that, first of all, there's a big increase of stroke incidence with age. And secondly, there's a difference for men and women. So 1 in 5 of the women listening to my talk are going to have a stroke, but it's only 1 in 6 of the men. And that's because men die earlier doing other stupid things like smoking, riding their motorbikes too fast, you know, the kind of thing. Now, most strokes involve a clot blocking an artery, which you can remove. If you're rushed to hospital, they will remove the clot from the artery and they'll say, job done. But it turns out the capillaries often do not reperfuse. This was shown by Adelbert Ames back in the 1960s. He did a classic experiment where he injected India ink into the blood of a rat, I think, and he showed that in control conditions, the India ink goes through all the blood vessels. But if you then do middle cerebral artery occlusion on the animal and then reperfuse the artery for an hour, and then inject the India ink, you find that there are lots of non-perfused regions then, as you can see in this diagram on the right here. So although the large vessels reperfuse, some capillaries, many capillaries, fail to refill. So what's going on here? Well, in the retina, we looked at the role of pericytes in this by applying ischemic solution. We found that in ischemia, the diameter of the capillaries near the pericytes goes down, as you can see here. And that's not surprising because normally pericytes use ATP to maintain calcium pumping out of the cell. And so when you have a stroke and there's no ATP, that calcium pumping stops and the calcium concentration rises, and that will activate the myofilaments and that will squeeze the capillary, okay? But that doesn't explain why it's so long lasting. You would think that when the blood comes back, all of that would be reversed, but it isn't. Incidentally, this also occurs in vivo after middle cerebral artery occlusion. This is in a mouse expressing a red dye in the pericytes again, and there's green dye, phythialbumin, in the blood. And we've labeled the vessels with isolectin B4 conjugated to this purple dye. And you can see that after a middle cerebral artery occlusion for 90 minutes and reperfusion, the green blood comes back through lots of vessels, but there are some vessels, like these ones up here, which are still present, they're purple, but there's no green blood in them. So what's going on there? Well, we can find out what is going on there by drawing lots of little lines across the vessel like this, using a semi-automated procedure called a PhD student. And you draw all these lines. They say it's really enjoyable. And you can then measure the diameter of all these vessels. And you find that in the core of the lesion and also in the penumbra, there's a decrease in the diameter of the capillaries, which is larger near pericytes. There's also occlusion of vessels by the blood just gets clotted there, or it gets blocked by a neutrophil. So it seems as though pericytes constrict these vessels. And this is consistent with earlier work by a Turkish group with Müge, Miski, and Turkey Dalkara in it. So if you look at the blood flow to the brain in this kind of situation, when you do middle cerebral artery occlusion, the blood flow stops. You then open up the artery. This is what would happen if you're rushed to the hospital and they remove the clot from your middle cerebral artery. The blood flow comes back transiently. There's some reactive hyperemia, but then it settles down at about 60% of the original level. And this is very long lasting. It goes on for hours. And so the question is, why is it so long lasting? Because this contributes to the death of neurons. So we think we understand this, and we've got the answer by imaging in brain slices. So here you can see capillaries perfuse for a long time in normal solution. You find they maintain their diameter for a long time. But if you apply ischemic solution, this is chemical ischemia, blocking ATP production both by glycolysis and by oxidative phosphorylation, then you can see these little red lines. The diameter of the capillary gets smaller and smaller, as is plotted just here. And the question is, why doesn't it come back afterwards? Well, if you then, after an hour, apply propidium iodide, which is used to measure the integrity of the cell membrane, you find that after an hour, the membrane has either ruptured or become very, very permeable to large ions like propidium, and this goes in and labels the genetic material. So you can see that essentially the membrane has broken down and the cell has died. So we think what's happening here is there's a constriction of the myofilaments in the pericytes, but then the cell dies in rigor. Okay, if you remember your muscle 101, you need ATP to relax muscles, not to contract them. And this is why corpses are stiff as well. So what's happening here is they constrict, then there's no ATP, but then the cell dies, so then they're locked in a constricted state and no more blood flow. Well, blood cannot flow so well through the constricted capillary. This is just a graph of the constriction occurring over about 15 minutes and the death occurring. In chemical ischemia, it only takes about an hour to kill all the pericytes. They're very sensitive to death, to ischemia. It's slower actually with middle cerebral artery occlusion in vivo because that's not such a severe insult. So that was in the gray matter. The same thing happens in the white matter as well. So we think that this rise of calcium, which occurs in ischemia, kills the cell. And that's why there's a long-lasting decrease of blood flow. And this means that this long-lasting decrease of blood flow here could reflect the death of the pericytes. And it means that pericytes are a therapeutic target in stroke. One of our aims now is to try to reduce this death and long-lasting constriction mediated by the pericytes. And in fact, in the paper we published on this in 2014, we showed that if you remove the calcium, you can slow this, this constriction and death. So blocking voltage-gated calcium channels might be one approach. Okay, I now want to turn to another issue, which is the role of pericytes in Alzheimer's disease, where they turn out to play a similar role. But first, I want to have a digression onto the properties of the immune cells of the brain, the microglia. Many of you will be familiar with microglia, I guess. They look like this. They've got these processes which constantly survey the brain. This is a 30-minute loop here, and labeled with isolectin B4, this microglial cell is. And if you binarize this image to turn it into a black and white image, and then add all the images together to see everywhere it's been in the last 30 minutes, you can see that over an hour or two, this is going to scan out all of the region of the brain around it. And this scanning is important to detect dead objects that need to be removed, to prune redundant synapses during development and so on, or to remove amyloid beta. Now, whilst microglia are doing this, they interact with lots of other cells. So here's an astrocyte in red, and this is a microglial cell having lots of interactions with this astrocyte. This is from the work of Nimian 15 years ago. They also wrap their arms around little neuronal somata, and in fact, they've been shown to calm down the firing of neurons. And for the purposes of this talk, the important thing they do is they interact with capillaries, as you can see here. Now, why might this matter? Well, as I've shown you, nearly 60% of the vascular resistance in the brain is in capillaries. And secondly, microglia synthesize vasoactive agents. And if you look at transcriptome data, they are the one place in the brain which synthesizes thromboxane A2. They also generate endothelin-1, which is a powerful vasoconstrictor. And in pathology, they can also generate cyclooxygenase derivatives like prostaglandins. So they do all of those, they generate all of those things. And it's known that after ischemia, microglia extend processes towards blood vessels. So we wondered whether the same thing happens in Alzheimer's disease. And to do this, we looked at a particular model mouse for Alzheimer's. It's called the APP-NLGF mouse, which has got mutations. It's a knock-in mouse, so the endogenous mouse APP has been replaced by a human APP with various mutations which increase the production of amyloid beta and increase its aggregation. And we can then look at how far apart microglia and pericytes are in wild-type animals and in the AD mice, and we find they get closer together. So that may indicate that these immune cells could have a bigger effect on blood flow in Alzheimer's disease than they do in the wild-type mouse. Now, why should I say all this? Well, it turns out that if you survey a large number of biomarkers, what you find is that the first thing which changes as you go from being healthy through mild cognitive impairment to having late onset AD, Alzheimer's disease, the first thing that changes is blood flow. What this graph shows in brown is the decrease of blood flow which occurs as you develop Alzheimer's disease, minus the change which occurs due to aging. So old people like me have got less blood flow than some of the young people in the audience, I hope. But that change has been removed. And so this just shows the decrease of blood flow due to the disease. And this takes off before you start getting deposition of amyloid beta or phosphorylation of tau or loss of memory and so on. So this suggests that there may be a big vascular component to Alzheimer's disease. And interestingly, about the time that the amyloid hypothesis was proposed in the early 1990s by John Hardy and colleagues, some Japanese workers showed that if they ripped blood vessels out of the human cortex, they found that there were characteristic squeezings of the blood vessels, a bit like the ones I've shown you are produced by pericytes. And in more recent work, Leif Østergaard has shown, using imaging techniques, that there are disturbances in the control of capillary blood flow in both human and mouse models of Alzheimer's disease. And what he does is to measure the capillary transit time in the brain, for example, in humans. The transit time tells you how long it takes the blood to get through the capillary once it's in the brain. And of course, that gets longer when the blood flow goes down in AD. But in addition, the heterogeneity of that transit time goes up. And they attribute this to changes in the degree of constriction of capillaries by pericytes. So there's a change of capillary control of blood flow in early Alzheimer's disease. And this, importantly, this correlates, this increase in heterogeneity of the transit time correlates with cognitive decline as measured by the mini mental state exam. Okay, so we wanted to look at control of blood flow by human pericytes in Alzheimer's disease. And to do this, we started out by teaming up with a neurosurgeon who routinely has to remove bits of the human brain in order to get to gliomas, which are deeper in the brain. And so we wait in the operating theater there, and we get given the supposedly normal brain, which is on top of the glioma, and we then cycle it round to the lab, and we make it into brain slices like we do for a mouse. This is a piece of human brain. You can see the pericytes on it here. They label for PDGFR beta, a classic pericyte marker, and they also label for alpha smooth muscle actin, which is one of the contractile proteins which produces the constrictions I'm going to show you, at least we assume it is. And if you look at these blood vessels, this is now a live, that was histology, but this is a live human brain slice. You can see a pericyte here, and we apply noradrenaline to it, and you can see that it squeezes the blood vessel near the soma. Here it is decreasing the diameter. If we apply glutamate, it dilates. And so the conclusion is that human pericytes, just like mouse or rat ones, regulate capillary diameter near their soma. Now, why do they do it near the soma? It's near the soma because that's where the capillary puts circumferential processes around the vessel. You can only change the diameter if you've got processes going around the vessel. It's no good if they just go along the vessel. And so I counted the number of circumferential processes as a function of distance from the soma. And it turns out that 80% roughly of the circumferential processes are within 10 microns of the soma. So if you take a brain slice like this from the human and apply U46619, this thromboxane analog, you find you get a big constriction near the soma, but it's a smaller constriction as you go away from the soma. So in control conditions, the diameter is actually larger near the soma, and that's because the pericyte induces growth of the endothelial tube. But, so this is diameter and this is distance from the soma. But once you've added the vasoconstrictor, the constriction is largest at the soma and smallest away from the soma. And so you just get the largest percentage constriction near the soma. I want you to remember this middle graph here because we're going to come back to that many times. So what's the relevance to the control of blood flow in Alzheimer's disease? Well, if you take this brain slice and make amyloid beta oligomers, which are thought to be the neurotoxic species in AD, you find that when you apply them, that induces a slowly developing constriction of the capillary near the pericyte soma. You can see this happening in a human brain slice here, and it also occurs in rat and mouse brain slices. So if I play this movie, you can see that applying A-beta leads to a constriction of the capillary. You can pick off stills from this kind of movie and plot the diameter over time, and you see that amyloid beta 1 to 42 or 1 to 40 both constrict the capillary, whereas a scrambled version of A-beta doesn't. And importantly, the amount of A-beta you need to do this has an EC50 of about 5 nanomolar, and Colin Masters' group has shown that in human AD, the concentration of soluble A-beta, that's the oligomers, in AD brains is about 6 nanomolar, so it's in the right ballpark. Of course, this is a stupid experiment, because here we're trying to mimic a disease which takes decades to happen with applying amyloid beta for an hour to a brain slice, but I'll come back to that later and show you that it is relevant anyway. So how does it work? Well, there's a whole slew of proteins which have been suggested as receptors for amyloid beta. When the A-beta oligomer binds, it's proposed to activate NADPH oxidase in certain cells, either microglia or endothelial cells or pericytes. And that, interestingly, if we block NADPH oxidase with this drug diphenyliodonium, which isn't very selective, but anyway, then we block this constriction of the capillary. This NADPH oxidase generates electrons, which then make superoxide. And if we superfuse superoxide dismutase to scavenge reactive oxygen species, that also blocks the constriction. And finally, if we block endothelin receptors, which have been suggested to be activated in these kind of situations, that also blocks the constriction. So we believe that the superoxide or some other reactive oxygen species activates endothelin release, which then acts, all of these cells express endothelin, and that then acts on endothelin A receptors on pericytes. Okay, so this is,
yeah. Question here. Do you know which species of alpha-beta are there in the 6 nanomolar concentration applied to human slices?
Yeah, I showed you which species of A-beta. Is that what you're asking?
Yeah.
Okay, so I showed you a little blot here. So this is the A-beta 1 to 42. These are the monomers down here. The molecular weight is about 4.5. So up here you've got, it's about 4 and 5 times the molecular weight is probably the species which is acting.
Okay.
Okay?
Yeah.
So why do we suggest that it's happening in this order? Well, if we apply hydrogen peroxide to generate reactive oxygen species, that also generates a constriction. And this is partly blocked by the endothelin receptor blocker. So we assume endothelin is downstream of reactive oxygen species. But if we do it the other way around, applying endothelin, then when we apply superoxide dismutase, it has no effect. So we think it occurs in this order. And by doing other pharmacology, it turns out that it's mostly NADPH oxidase 4, which generates the reactive oxygen species. So which cells is this in? Well, if we use imaging of reactive oxygen species, this is work done by Pablo Izquierdo, who is watching this seminar, so I have to mention him as I'm not sure I have his name on the slide. So you get more reactive oxygen species produced. And then if we image these in greater magnification, you can see that the cells generating the reactive oxygen species co-localized with a pericyte marker are not very well with microglia. In fact, we've discovered that both microglia and pericytes generate reactive oxygen species, but in the short term, it turns out that it's mostly pericytes. It may be microglia more in the long term. So A-beta constricts capillaries via pericytes, but we wanted to know whether this occurs in Alzheimer's disease itself. And so to do this, we teamed up with neuropathologists who were able to provide us with human cortical biopsies. This is not a common procedure, but it turns out that at the hospital associated with our university, if you happen to be dementing for no known reason and they think there may be a cause they could cure, they will happily remove part of your brain while you're still alive from a part of the brain which they claim doesn't do much anyway. It's the right prefrontal cortex. And then we can look at these bits of brain, and what they use that for is to see if there's A-beta deposition or not. Roughly half the patients have no A-beta deposition and half do have it. And then we can label the pericytes using antibody to PDGFR-beta and look at the diameter of the capillaries, okay? And when we do that, if you measure 9,000 diameters on these patients' brains, you find there's a small decrease of diameter. That doesn't sound very much, but an 8% decrease, if it was uniform, would reduce blood flow by 30% from Poiseuille's law. But we want to know whether this is non-specific or due to pericyte constriction. And it's interesting that Seth Love's group and other groups have shown that endothelin levels are raised in AD brains. And I showed you a minute ago that endothelin is part of the chain of events leading to this constriction. So to pin this on pericytes, we simply measure the diameter as a function of distance from the pericyte. And when we do that, in the patients without A-beta deposition, we find the diameter is larger near the soma, which is what I showed you before in brain slices. Whereas in the ones who are depositing A-beta, there's a big constriction at the soma. Okay. So it's pericytes which are constricting these people's blood vessels and reducing their blood flow. This is the slide I showed you earlier with the same thing in a brain slice. So this predicts a 50% decrease of blood flow with simple estimates. which is roughly 45% is what's seen in human patients with AD in the affected regions. Now, we can look at how this varies with patients with varying amounts of A-beta being deposited by looking at the extrapolated diameter here at the pericyte soma on a case-by-case basis. And that diameter is found to decrease rapid. Each point is one patient. It decreases rapidly as you start to get deposition of A-beta. So this loss of blood flow is predicted to be an early event, which it is. So the constriction occurs early in AD. We sent this to Nature, and one of the kind Nature referees says it's all very well doing this in humans, but what about doing it in a transgenic mouse? So we did, and you find the same thing. There's a constriction of the capillaries. It's larger near the soma, and again, you get this variation with distance from the soma. In the mouse, you can look in the cerebellum, which doesn't have amyloid beta plaques, and there you see there's no constriction at the soma. So there's a tie-in with A-beta there. And in addition, exogenous A-beta has been reported to constrict arterioles, but at the levels of endogenous A-beta produced in the mice, there is no constriction produced. So it's not being produced by arterioles constricting in the mice, and perhaps not in the humans. Now, another group, Chris Schaefer's group, has suggested that neutrophils occlude capillaries in AD, and they find an increase in the number of blocked capillaries from 0.4% to 1.8%, which is predicted to reduce blood flow by 5%. We would imagine that this constriction by pericytes is going to make it easy to trap neutrophils in the blood vessels like this. But nevertheless, this predicted effect on blood flow here is much smaller than the predicted effect if you simply consider the blood as a fluid without cells in it. So we think simply the increase of resistance is the bigger effect. Okay, can we prevent or reverse this constriction? I've shown you that this is what's happening. A-beta is dumping calcium via endothelin receptors, and that constricts the capillary. How can we stop this? Well, we could try blocking the NOX4 or the endothelin receptor, but endothelin receptor activation is fairly irreversible pharmacologically. It doesn't work very well. So we also used another strategy where we apply C-type natriuretic peptide, which raises cyclic G, and that stops calcium release from internal stores. And also via a Viagra kind of mechanism, it activates myosin light chain phosphatase to remove the phosphate and decrease the contraction. So that's the diagram I've just shown you. When we induce constriction by A-beta in a brain slice, this is what normally happens. If we block these activating phases here, you stop any further constriction occurring, but it doesn't reverse very quickly. But if we apply the C-type natriuretic peptide, then that gives you a rapid reversal of the constriction. So we're now working on further developments of this to try to stop the constriction of capillaries in AD. And I just want to sum up what I've shown you about AD. So A-beta constricts capillaries, blood flow goes down, oxygen and glucose levels fall. And this reconciles genetic evidence for the importance of A-beta with the fact that the earliest event in AD is a vascular dysfunction. I've shown you some mechanisms we could use to block this. Importantly, other people have shown that when energy levels fall, that upregulates the enzyme which makes A-beta. So there's a positive feedback loop here. And this implies that if for some other reason you get transient ischemia, you may kick off this positive feedback loop. So people who have sleep apnea, for example, or who have repeated blows to the head, boxers, they are known to get a decrease of cerebral blood flow, and they're also both known to have an increased chance of getting Alzheimer's disease. And that may be because of this effect on pericytes. A-beta eventually leads to hyperphosphorylation of tau and synapse and neuron loss, but hypoxia and loss of glucose will also do this. And so it's an open question now, I would say, what fraction of the loss of synapses and neurons is produced directly by the A beta and tau, and what fraction is produced by the hypoxia, which the A beta produces. So summing up, these are the people who've done the work, a vast number of them. This is the famous Pablo I'd like to come back to. And there have been collaborations with many other people. Thank you for listening, and I'll answer any questions. I think you'll need to unmute people. We can't hear you, Jorge. I can hear some children though. Okay. Anyone want to ask a question? I think Jorge.
Oh, hi, David. Do you hear me?
I can hear you. I can't see you, but I guess I don't know. Yes.
Thank you for your great talk. I want to ask you a question. It is regarding if you studied whether A beta and U46 have an accumulative contraction effect on capillaries.
U466 is a dilator. So we haven't, the experiments with A beta were not done in the presence of U466 because we didn't need to. The A beta constricts on its own. The U466 dilates. Oh, sorry, I'm totally confused, aren't I? Let me start that whole answer again. They do both constrict. Yeah, sorry.
Yes, okay, yeah.
I should have had more coffee.
Don't worry.
That's better. Yes, of course, U466 is a constrictor, but we didn't apply the U466 during those experiments. It would be easy to do, but we haven't done it. Are you suggesting that maybe U46 thromboxane A2 is released during AD as well?
Oh, yes. Well, I was curious whether if it was altered, the contraction effect of maybe they act through the same mechanism, and so A beta, basal A beta in AD is like predisposing or something like that the capillaries to have a further contraction.
Yeah, I mean, eventually these two mechanisms are going to occlude each other anyway, because pericytes can completely occlude the vessel. And so you can't have more than 100% constriction. So it will just depend on how much the calcium rises. Yeah.
Okay. Well, thank you.
You're welcome. Sorry, I messed up the start of it.
Don't worry, it's fine.
Someone could ask me an even easier question.
I was like, okay.
Yeah. No, I was thinking about prostaglandins. Yeah, more questions.
Thank you. I enjoyed your question, your talk.
Thank you.
Okay, any other question? So I have one. I mean, have you tried GKT together with the injury, with the injection of A beta to kind of, or in a ischemia context? So that
Which drug? Sorry, the GKT.
GKT.
Yeah, we showed.
But not the GKT, the ligand for the receptor.
For endothelin.
Yes, no, for the receptor that you use for increasing the dilation.
You mean the CMP?
Yes.
Have we tried endothelin with CMP? No, we haven't.
Or CMP.
I would predict that it would stop the endothelin constricting, but we haven't done that because life is short. Yeah, but I mean, it ought to work because we believe the A beta is only working via the endothelin receptor. I would predict it would do the same thing. It will be easy. It will be easy to test. Yeah. Yes. Yeah.
The idea will be that
You need to, you need to unmute.
Oh, yes, you keep dilated the capillary.
Oh,
And
sorry.
you
Sorry, can you just ask your question again, Jorge? And then there is someone called Ria who wants to ask a question.
Okay, perfect. Yes, there's a lot of people trying to ask, I think. But yeah, the question is, with CMP, you get dilation.
Yes.
If you apply an ischemia, maybe you can keep the capillary dilated, and you will not have this death of the pericytes.
It's possible, but I assume that the rise of calcium during ischemia is due to a failure of calcium pumping rather than just a dumping from stores. But I mean, it may bypass it in this way. It's a very good idea. We haven't tested it though.
Okay, thank you.
Yeah. So maybe someone else was asking a question.
It seems that somebody cannot ask because it has blocked the microphone, but they haven't blocked everyone.
Can I do anything about that?
Ruth, can you ask now?
Can you unmute everyone?
Yes, I have done it. Okay, so strange.
Everyone still looks muted though.
Yes, I am the host. I will try. I mean, let's see if I can do something else. Activate everyone. I have deactivated all microphones, so I don't know where. I can try to see if I can find Ruth. She wanted to ask something, I guess. I cannot see her. Okay. I cannot find her. Anyway. Strange.
Okay, does anyone else want to ask a question? Raise your hand. There seem to be 31 questions
Yes,
in
there is one here.
in the chat.
Yes.
Should I
Yes.
should I just go down? Oh, the retina is good. Yes, I agree. Let me just go down the questions in the chat. Maybe this. Okay, so someone called Daniel has asked, what's the contribution of intracellular calcium to the contraction? We think it's almost entirely the main cause of the contraction, but it may also be that Rho kinase makes a contribution to the contraction as well, as it does in smooth muscle in general.
No.
Jorge,
Yes, Juana.
¿puedo?
Yes.
David, do you think that there is some problem with the astrocyte and the energy given to the neuron that can be participated in this problem with the capillaries or pericytes?
Well,
in Alzheimer's disease, I guess.
Right. I'm not aware of any evidence that it's specifically an astrocyte energy provision problem. You mean in terms of lactate transfer or something like that? I mean, as you probably know, there's a massive debate about how important lactate transfer from astrocytes actually is. But I believe that because the blood flow is the first thing to decrease, and it decreases by 45%, I think that's going to be the main cause, main initiating cause of the failure of energy supply. Just for calibration, if your blood supply decreases by 30%, then you stop protein synthesis. So clearly a 45% decrease is going to have a big effect.
Thank you.
Okay, Javier Ramos also has a question. You can unmute, yes.
I can speak. Do you listen to me?
Yes, I do.
Amazing, amazing talks. Thank you very much. I have a question that is, if the astrocyte is contacting directly the pericyte by the end feet, because the pericyte is touching the membrane, the basal membrane, and also the end feet of the astrocyte, so the contact is there, and the prostaglandins are released there, or if the astrocyte releases to the blood flow, and then pericyte detects the prostaglandin from the blood flow.
I don't think so. I think it's the first one. The basement membrane does surround the pericyte, and lots of anatomical kind of people get very excited about the basement membrane as though it's like a Berlin Wall around the cell. But I don't think it's like that at all. When we apply drugs to the pericyte, they diffuse to the cell membrane and have an effect straight away. So the basement membrane is not like a solid wall. It's just molecules like any other piece of gooey brain. So I think that the astrocyte would release the prostaglandins into the extracellular fluid, and that would then affect the pericyte. The end feet wrap around the pericyte anyway. Yeah.
Okay. Thank you very much.
You're welcome.
Francisco Barrantes also has a question. I don't know if he's here. He wrote it in the chat.
Shall I just go through the chat and answer all the questions?
Okay.
Can you see the chat on my screen?
No, but we can see them. Yeah.
Okay, so Matias Monteverde said, is it known whether CNP would produce relevant secondary effects in other cells if used in a therapy? Well, it's going to raise, it's going to change the cyclic nucleotide levels and suppress calcium release in all cells, I guess, that express its receptor. And so, although its receptors are relatively concentrated on pericytes, I doubt that it's specific enough to use like that. You would probably want to somehow target the drug to act specifically on the pericyte. Maybe it's just a fantasy, but maybe you could have a drug which has got two ends to it, one which binds to the CNP receptor and the other which binds to a pericyte-specific receptor like PDGFR beta, something like that. There's a lot of stuff about the microphone in the chat. Okay, do CNS, so Francisco Barrantes says, could pericytes play a role in the microthromboembolisms in COVID-19? This is actually something we're working on. Pericytes are very important for COVID because they express the ACE2 enzyme. This is angiotensin converting enzyme 2. For those of you who don't work on the renin-angiotensin system, that system controls body fluids by producing angiotensin 2, a peptide, which is a vasoconstrictor. And that peptide is then inactivated by this enzyme called ACE2, which converts it from being a constrictor into angiotensin 1 to 7, which is a dilator. When the virus binds to ACE2, is the enzyme, that means you're going to have a higher concentration of angiotensin 2 in the blood and a lower concentration of its dilator, the angiotensin 1 to 7. So you're going to get more vasoconstriction, and that's going to decrease blood flow, which does occur in COVID-19 in a whole range of organs, the heart, the brain, and the kidney. In addition, it may be that entry of the virus could raise the calcium concentration in pericytes and make them constrict the vessels more. But in addition, Christa Betz-Holtz has a paper on bioarchive. You can find it just by going to Googling Betzoldt's bioarchive, where he suggests that if pericytes are damaged by the virus, like the virus goes in and replicates and kills the cell when it releases the virus, then that has an effect on the surrounding endothelial cells, and it makes them release von Willebrand factor, which is a major pro-clotting agent. So it's speculated that the reason there are these emboli, this pro-clotting environment in COVID-19, is due to a high concentration of von Willebrand factor. And I know of several groups who have unpublished data showing that the people who die in COVID-19 after getting that have a higher von Willebrand factor in the blood. So the pericytes may well play a role, partly by constricting, but maybe more importantly, by dying and triggering a release of von Willebrand factor. Anya Vivinetto wants to know if pericytes turn over. I think they don't turn over very much under normal conditions because we've done a little bit of BRDU labeling. But when you have brain ischemia and a bit of the brain dies, then they do start dividing, and they then move into the infarct region and they initiate the production of new blood vessels. How does prostaglandin release change in reactive injuries after an injury? I would probably pass on that, but it's known that astrocytes become less able to activate dilating pathways after stroke. And it may be because they undergo more calcium transient activity. And there's some evidence that when the calcium goes higher in astrocytes, rather than releasing prostaglandins, they release another arachidonic acid derivative, which is called 20-HEAT, and that's a vasoconstrictor. I think I've got to the end of the questions on the chat.
There is one here from Matias Monteverde.
Okay, one just arrived. Diana Jerusalinski said, can this be connected with sexual dimorphism in AD? I thought you mean more women get AD than men. I thought that more women got AD simply because they live longer, but I may be wrong. In other words, it's not a greater tendency. I may be wrong. I'm not very strong on that evidence. Okay.
There is another one.
Annalise Moya said, are there any other CNS targeted viruses that affect pericyte function? There is another one which goes into pericytes, but I can't remember what it is, sorry. But I'm sure you can find it in PubMed if you Google, if you look for pericyte virus. Daniel says, are pericytes sensitive to PCO2 and PO2? They are. I have a student in my lab who's looking at the effect of hyperoxia and hypoxia, because often people are given oxygen for certain conditions in the health system. So if you have a heart attack or a stroke, it's quite common to give hyperoxia. And he's doing both experimental studies on that, which show that hyperoxia makes pericytes constrict vessels. And he's also doing a meta-analysis of the literature on the effects of hyperoxia. And it seems to be bad. So it may not be great to have oxygen blown up your nose unless you have a very low oxygen in your bloodstream. Yeah, and PCO2, yes, they dilate. That's it. I think I've got to the end again.
Okay.
Any other questions? Seems like we might have got to the end.
Yeah. Okay, David. Thanks a lot. It was
Okay.
amazing talk. Yeah, we have a lot to.
Thank you very much. Gracias de Londres. And maybe I'll see you in South America one day.
Yeah, hopefully.
Okay.
Very happy to have you here.
Okay. Thanks for the invitation.
Thank you.
Right. I should switch this off somehow now.
Yeah.
Right. Okay. Good. Okay.
Perfect.
Hasta la vista. Gracias.
Yeah, no. Muy bien. Perdón por los inconvenientes.